Let's look into the scoring calculation or scoring function in MSA. We look into the scoring functions. We'll repeat here the sum of paired scores and how these scores which are captured in paired are put into a scoring functions, and then in the end we will see that the overall score of the alignment is the summation over all individual scores for all pairwise comparisons. So to find an optimal alignment, it is important to measure how good is the alignment, and the criteria in order to get this goodness is actually the alignment score. So for that we try to use some scoring functions. So scoring should take into account these following facts. Number one, that there are some positions in nature they are more conserved than the other ones. We should also consider that sequences are not independent, but they are related by some phylogenetic histories which are being represented by phylogenetic trees. And then these alignments should maximize the possibility of finding the common ancestors. So all these things, including these phylogenetic relationships, they are kind of taken into account while scoring those alignments. So while we are doing these scores, we take those individual positions as columns. We do those multiple sequence alignments, and then we look into each nucleotide position as a column, where we have a column in which there are different nucleotides top to bottom coming from different sequences are put together. So we assume that these columns are statistically independent. So sum of the score for the whole column is equally that is given by this relation. So that is actually representing the score of the overall alignment. So SM is summation over all scores for all possible columns, where MI is column I of multiple alignments M. So here we are saying that the score of the alignment, overall alignment, is summation over scores of all columns. So we define M as the matched sequences or the alignments, and then we can observe two types of parameters that we can put into the score. So M, I and J were a symbol in column I for the sequence J. So for example, if we want to know about what is the fourth symbol in the second sequence, we see it's a G. So it's represented as blue color for your convenience. Same way, we can put the observed counts for residues. For example, for one of the residue which is A over here in column I, we can have it as Ci into A. For example, in column in C1A, how many A's are there in column number 1? So we see there are 2. And how many C's are there in column number 1? So we don't see any C's, so we put the 0 over there. How many G's we see in column number 1? We see it's only 1 right here. How many C's we see in how many T's we see in column number 1? We don't see any T, so it's a 0. And how many gaps we see? Again it's a 0. So in this way we can observe the counts for the residues, different residues or different nucleotides in each column. So let's go back to sum of pair scores of a multiple sequence alignment M. That is the sum of the scores for all individual pairwise alignments, and sum of score can be SP or sum of pair score for column I is given by this relation, where SMI is summation over S for a K and L, where we have K less than L. This score for each pair which is compared A and B, so A may be the first nucleotide in the first sequence and B can be the first nucleotide in the second sequence. So their score is actually derived from the substitution matrix. So as long as they are pairing up, you can come up with a match score. So if we have a substitution where they are not pairing up, you find a mismatch, so then you will consult this substitution matrix. And while we doing those sum of pair scores, here is another way which is there in your book, so where you can go and read this Mounts book, so where you can see that these alignments are shown, where we have 3 column alignments where we have 3 nucleotides in each of the sequences, and total we have 5 sequences. So if we go by each column, we can have these possible comparisons. So we can come up with a shape like a star. So where here we can have total 10 possible combinations. So and if we look into the column number 1, so they are all matching with one another, so you will get 10 alphas or 10 match scores. Let's move on to the next column. Here we see that there are 6 matches or 6 match scores, and then we have 4 mismatches. So your overall score is 6A minus 4 beta. So whenever we have a mismatch, we generally penalize for that, or we look into the substitution matrix where we can get some negative numbers. So in this way, a match score is always adding something to your overall score, whereas the mismatches, they are subtracting from the overall score. So in the second alignment scenario, in the second star, you will see that you have 6A minus 4B. And in the third situation, you have 4 matches and 6 mismatches, which was actually in the third column. So you come up with your scoring schemes, whatever you have defined, you put into your matrices, but overall relations or the formulas, they go like this. And what about the overall alignment score in this case? So that is, we have 20 alphas, 20 match scores minus 10 betas. So that will give you the overall score for this alignment. So I guess with this example, it has been more clear that how this sum of pair scoring is done. So in order to find the optimal alignment, multiple sequence alignment, it's important to measure how good is the alignment, and for that we need a scoring scheme.